Good afternoon to all distinguished professors and fellow researchers. My name is Youngmin, a third-year PhD candidate at Foundation University in South Korea. Today, I will briefly introduce my plant systematics research conducted so far. I also hope to discuss the role and importance of systematics on conservation biodiversity. Traditional taxonomy is the science of classifying organisms based on their characters. This involves four main activities such as identification, description, classification, and nomenclature. These activities form the foundation for collecting, cataloging, and retrieving information on biodiversity. The modern systematics integrate data from ecology, morphology, genetics, cytology, and nearly all biological fields to comprehensively understanding the taxa and evolutionary phylogenetic relationships. Studying systematics and plurality research, we can evaluate the species vulnerability and make decisions on conservation priorities under resources constraints. Studying speciation mechanisms and biogeographic origins across the multiple countries, we can help inform the effective response to climate changes. I completed my master's degrees at Chungnam National University in Daejeon City, South Korea, where I conducted phylogenetic analysis on Korean yuccus. The yuccus is one of the most recently diverged lineage within the Jungkook family yuccus. It's an aquatic herbaceous plant typically found in wetlands, along riversides, agricultural fields, and coastal regions. In this study, I focused on cytotaxonomical and micromorphological analysis to investigate the evolutionary patterns and taxonomical useful characteristics. So over the course of 2 years, I conducted extensive plurality works across the South Korea. First, the Korean yuccus species exhibited a frequent polyploidy, including the triploid and tetraploid. This suggests that these ranges they have undergone rapid speciation and possess a high degree of adaptability to environmental changes. Polyploidy often arising from the unused gametes either through interspecies and interspecies events, leading to speciation and phenotype changes. For example, the Gigas effect enlarged cell size may occur, and low-energy expressions can enhance tolerance to drought, salinity, and or high temperatures, potentially leading to niche expressions. And the term karyotype refers to a number, morphology, and size of the chromosomes, and these traits can be a useful taxonomical traits when distinct differences exist among taxa. I was able to consolidate the findings into a research paper which was published in 2022. To identify additional taxonomic characters, I conducted micromorphological studies of leaf, stem, tepal, and pollens. While tepals and pollen showed little variation, but leaves and stems exhibit diverse taxonomically useful characters. For example, subgenus yuccus shows exhibit long epidermal and guard cells and lower guard cell densities compared to subgenus Agathria. So these traits help distinguish two subgenera and may reflect adaptations to specific environmental conditions. Compared to the moisture-loving taxa subgenus yuccus, the dry and coastal adapted subgenus agastrium likely evolved smaller and denser guide cells to minimize water loss and enhance photosynthesis. After completing my master's degrees, I worked as a postmaster's researcher at Korea National Arboretum for 3 years, and conducting plant conservation research in the DMZ and North Korea regions. As the world's only divided countries, the Korea's DMZ have formed a unique ecosystems over the past 70 years due to restricted human access and military uses. The DMZ is one of the 3 major ecological corridors on the Korean peninsula, harboring a diverse array of wildlife and spanning across 10 administrative districts. The western part of DMZ features low hills under 500 meters in elevation and wetlands and coastal regions. And the central region of DMZ features broad plains, and the eastern DMZ is dominated by Ekudega mountainous range, which exceeds 100 meters in elevation. So I conducted extensive forestry researches on explored mountainous areas in DMZ, and confirmed and presented conservation from the findings at conferences such as the 2021 World Forestry Congress. By compiling and cleaning data from all available specimens or literatures, we confirmed the presence of 1,830 plant species around the DMZ area, and published both checklists and research papers. That rare and endemic plants is dominated in eastern DMZ, and the alien plants is concentrated in urbanized west. Additionally, by reviewing North Korea floras and synonymizing Maine, we identified 486 taxa distributed exclusively in North Korea, not found in South Korea. And we assessed the status of genetic resources of North Korea using data from BCCI, GBI, POW, and global herbarium databases. And furthermore, we updated the North Korea genetic resources held by Korea National Arboretum on GBI app. Additionally, we conducted genetic diversity of rare plants distributed throughout the DMZ, and categorized the plants introducing plants difference by administrative districts. And we promoted northern region plants and North Korea plants throughout the wetlands and participation in 2023 Gangwon World Forest Expo. Through these efforts, I realized that the knowledge of our native plant species is very limited, such as taxonomy, distributions, evolution pattern is very limited. So, therefore, I began my doctoral studies in plant systematics at Gangwon Regional University in 2023 to further contribute to this field. Currently, I'm engaged in various activities such as forestry research and seed collection in Ekudega mountainous areas, while my doctoral dissertation focused on the phylogenetic and biogeography study of the genus Eplovium in Onagrassy worldwide. These groups distributed worldwide, predominantly across the northern hemisphere. It has four petals and disintegrating pollens, like capsules, and the seed-bearing coma. However, previous molecular researches suggest that the hybridization and incomplete range sorting as a major causes of polytomy and phylogenetic discordance. These suggest a complex evolutionary history with the effluvium, highlighting the integrating surveys using various characters. So I have conducted the various taxonomic researches using micromorphological characters, anatomical, and micromorphological characters. And I also conducted cytotaxonomical researches confirming the diploid chromosome numbers as 36. Moving forward, I intend to focus on morphological phylogenetic researches. My primary objective is to build a robust phylogeny using nuclear single copy genes and complete trust from next generation sequencing based on collection worldwide. And I will compare phylogenies and perform IRD tests to converse between nuclear and plasmid-based trees. If low phylogenetic converse is detected, I will perform hybrid tests to evaluate hybridization as a potential causes. Furthermore, to infer robust phylogeny, I will apply multi-species forest model analysis. Finally, I will estimate the divergence types using fossil data and apply models like DeepLike to classify speciation scenarios. Through this process, I enhance my ability to resolve phylogenetic relations, origins, and speciation mechanisms in plants with complex evolutionary histories. That concludes my presentations. I sincerely thank you for your attention.
Any questions? Have 3 minutes left. Okay, if there are no questions, once again, thank you so much. Thank you.